Welcome back. Next, we have Sigyn Therapeutics, Inc. It trades on the OTCQB under the symbol SIGY and is a development stage medical technology company headquartered in San Diego. The company's therapeutic candidates are each designed to overcome a clearly defined limitation in healthcare. Please welcome its CEO, James Joyce. Welcome back to the conference, James. How are you doing today?
I'm doing good, Anna. Nice to see you.
Great. The floor is yours. Call me back when you're ready for questions.
All right. I appreciate it. I'll talk to you soon. Prior to getting started, I want to point everyone out to our forward-looking statements, which we have published for your review on our website, SigynTherapeutics.com, under our presentations button. With that said, at Sigyn Therapeutics, we are designing devices to optimize cancer therapies and address untreatable infectious disease disorders. We launched our endeavors during the pandemic, and we now have an expansive lineup of therapeutic candidates that are based on 12 patents pending in the US and abroad. A distinguishing factor about our devices is that we've designed them from the ground up to provide potential strategic competitive advantages within the pharmaceutical and dialysis industry. In regards to our cancer products, which cancer is the #2 cause of death in the United States, we are focused on improving the delivery of therapies and also to reduce the toxicity of therapies. And I think you'll learn in this presentation, or you may be shocked in this presentation, that leading drugs that treat cancer are poorly delivered. In response, we develop chemo prep to improve the delivery of chemotherapy. We develop chemopure to extract off-target chemotherapy that's left in the bloodstream to reduce toxicity. We created immune prep to improve the delivery of immunotherapeutic antibodies, which are the top-selling drugs in oncology. And our lead product, Sigyn Therapy, we designed this to address infectious disease disorders that are not treatable with drugs. And what you're going to learn is that a majority of infectious disease disorders are not treatable with drugs, at least those that are infectious to humans. In infectious disease, I want to point out that we have relevant industry experience. I previously oversaw the development of the first medical device to receive FDA approval under emergency use authorization to treat an infectious virus. In this case, it was Ebola virus. This was also the first technology to receive 2 FDA breakthrough device designations. As the lead author of those regulatory submissions, I'm very proud of that achievement. And the technology played an instrumental role in expanding our government's definition of a pandemic treatment countermeasure to be inclusive of medical devices. Prior to this device, a countermeasure was defined solely as a drug or a vaccine, but thanks to the efforts of Joe Lieberman and his chief of staff, Chuck Ludlam, and these individuals were really the fathers of Project BioShield, which launched the industry for treating pandemic threats and potential bioterror threats. Thanks to their efforts, they had me march around Capitol Hill and testify before Congress on the use of medical devices to treat infectious viral pathogens. And this definition got approved. And as a result, in the subsequent pandemic outbreak of 2020 with COVID-19, the first 4 therapies approved under emergency use authorization were blood purification technologies. As a result, Time magazine named this technology a top 25 invention and a top 11 medical breakthrough. And I bring this up because, with the exception of our CFO, all the members of our Sigyn team worked by my side to advance this technology. Our lead product is a next generation technology called Sigyn Therapy, again, to address untreatable infectious disease disorders. Here's an image of Sigyn Therapy in action in animals, first in mammal studies at University of Michigan. The technology has some attributes that we think are unrivaled. First, we need easy deployment. In that regard, we designed it for use on dialysis and CRRT machines already located in hospitals and clinics around the world. It's highly efficient. Its optimal blood flow rates are 350 mils a minute, which means for the average sized person, the entire bloodstream can be processed approximately 15 times through the device during a 4-hour treatment. And it has immense capacity. It incorporates a formulation of adsorbent components with approximately 200,000 square meters of surface area on which to capture therapeutic targets. Now, to put this in perspective, 200,000 square meters is the equivalent of approximately 50 acres. So consider 50 acres of absorption capability in the device. We validated its broad spectrum capabilities in vitro studies with human blood plasma. It has the ability to clear a wide range of viral pathogens plus bacterial toxins, including endotoxin. And it has the ability to address a broad spectrum of pro-inflammatory cytokines. Based on these capabilities, its therapeutic opportunities represent some of the most significant unmet needs in global health. Again, untreatable viruses. In this case, of the 219 viruses that are known to be infectious to man, most of those are not addressed with an antiviral drug. And based on the continued global warming, the convergence of urban crowding and worldwide travel, 3 to 4 new viruses are being discovered each year. There's also an opportunity to address antibiotic-resistant bacterial infections, which is an emerging global health threat, endotoxemia, which is a primary cause of sepsis. And we're going to talk a lot more. This is part of our lead indication. And then sepsis itself, which is a leading cause of hospital deaths in the United States. Again, all of those indications are not treatable with drug therapies. So our early clinical and commercialization opportunity is in end-stage renal disease patients. These are dialysis patients. They're dependent on dialysis for survival. There's about 550,000 of these individuals in the United States. Their annual mortality rate is approximately 20%. That means on average, once these patients are on dialysis, they live on average for about 5 years. Endotoxemia, which I referenced previously, and concurrent inflammation underlie cardiovascular disease, severe infections, and sepsis, which are the 3 leading causes for shortening the lives of ESRD patients. And extending the lives of those with ESRD may offer a strategic competitive advantage within the dialysis industry. I wouldn't say may, I would say it absolutely would create a strategic competitive advantage within the dialysis industry. Value of extending ESRD patients' lives can be quantified. And by not just the value of improving their quality of life, but the actual business proposition for the dialysis industry, which in North America is dominated by 2 organizations, Fresenius Medical Care and DaVita. Fresenius has about 208,000 ESRD patients in their North American network. And based on this patient population, if you were to extrapolate the value of extending the lives of patients in their network by 1 month, each month is worth about $1.2 billion in additional revenues. These patients of theirs also spend a lot of time in the hospital each year. And when they're in the hospital, they're out of network and the dialysis companies lose revenues. So for each week of reduced hospitalization for Fresenius, that could result in $300 million in recouped revenues. DaVita has approximately 200,000 ESRD patients in their North American network. Based on each month of increased or extended life for their patient population, that values is an additional $960 million per month. Reduced hospitalizations for each week, that's worth about $240 million in recouped revenues. So our ESRD clinical plan, we've drafted an investigational device exemption for submission to FDA to initiate first-in-human studies. It calls for the enrollment of 12 to 15 ESRD subjects with endotoxemia and concurrent inflammation. This will occur at 3 clinical site locations. We've identified 1 in Hartford, Connecticut, San Antonio, Texas, and Las Vegas. Our device will be administered during regularly scheduled dialysis sessions. Now, consider this protocol where the dialysis patient is sitting in their seat 3 times a week for 4 hours of treatment, receiving dialysis. Consider that our device fits in series with their normally scheduled dialysis treatment. So nothing changes for the patient. And this is a modular feasibility study, which means we utilize this to demonstrate safety, but then we can bridge off of this in the pivotal studies for Class 3 medical devices that go through CDRH at FDA Pivotal studies are what's required for market clearance. And so we can bridge into pivotal studies for both ESRD patients as well as non-ESRD indications. Something really cool about this modular feasibility design is that in this study, we'll enroll the patients, and for the first week, 3 treatments, 4-hour treatment, it'll solely be their dialysis treatment, and we can measure and quantify their normal response and changes in blood chemistry resulting from normal dialysis. And then in week 2, treatments 4 through 6, we include Sigyn therapy in series, and we can quantify measurable differences in reducing the presence of endotoxemia and pro-inflammatory cytokines. And that's a very valuable data point that you normally can't collect in a traditionally styled feasibility study. There's also an emerging opportunity in kidney transplantation, another way to extend the lives of ESRD patients. In fact, the best way to extend the lives of ESRD patient population. However, there's 90,000 individuals on the kidney transplant list, and there's a major shortage of organs available for transplantation. Each year, approximately 25,000 transplants occur, and as a result, many ESRD patients die before they ever find a matched kidney for transplantation. As a result, there's an emerging field of xenotransplantation that's driven by the major shortage of organs and the evolution of gene editing that's allowed for animal species, specifically porcine species, pigs, to be able to emerge to have organs that are compatible with human organs. And the leading area in this is related to kidney transplantation. So on October 15th, we filed a patent entitled Extracorporeal Therapies for Xenotransplantation. And we spent a lot of time studying FDA's guidance on xenotransplantation, and their major concern is to mitigate the risk of a pathogen that could be transmitted from a porcine organ into a transplant recipient. And not just the fear that it would cause potential rejection of the organ, but the potential for it to infect the transplant recipient and establish the ability to be transmitted in host-to-host infections in humans in general. So we looked at that guidance, and we looked at the capabilities of Sigyn therapy, and we believe it can be offered as a means to treat gene-edited pigs to reduce pathogen accumulation in their donor kidneys before they're extracted for human transplant. And again, this corresponds with FDA guidance. And to date, you might be surprised to know this, we've already administered Sigyn therapy to 8 pigs so far. The other opportunity we see is the treatment of human transplant recipients to extract pathogen and other circulating factors that could induce organ rejection. So again, not only to reduce or mitigate the risk of a pathogen from the pig donor to infect the human, but this is also viable in human-to-human transplants, because these patients, when they receive their transplant, are on immunosuppressive drugs, which often allow for the activation of latent viral pathogens, such as Epstein-Barr virus, herpes simplex virus, that many people carry around and can trigger organ rejection. So the interesting thing about this emerging field is that if there's an increase in transplants in ESRD patients, that would reduce the patient population for the dialysis industry. However, I'll point to you, there's 2 players that have emerged as leaders in this field for creating human-compatible kidneys. One is United Therapeutics, which just announced the building of their second $100 million facility to support these types of endeavors. And the other is eGenesis, who last month announced the completion of a Series D financing equal to $191 million. But I'll point out to you that the lead investors in this financing included Fresenius Medical and DaVita. With that said, our opportunity in oncology is significant. These are earlier stage developments. And I want to make it clear that we don't develop drugs to treat cancer. We're developing devices to improve the benefit of drugs to treat cancer. Related to chemotherapy, this is the most common class of drug to treat cancer, but peer-reviewed published articles would indicate that less than 3% of the drug is delivered to its tumor cell targets. And a majority of chemotherapy that's delivered is off target, and this is what contributes to toxicity. We are targeting to reduce the circulating presence of tumor-derived exosomes, which interfere with delivery and are associated with treatment resistance. Now, you have to understand that in individuals that don't have cancer, there isn't a presence of tumor-derived exosomes. But in those that do have cancer, the presence of these particles correlate with disease progression. And as published by Dr. Raghu Kalari at MD Anderson Cancer Institute in Houston, the blood of cancer patients is estimated to contain about 4,000 trillion exosomes. That's 4 quadrillion. That's 4 with 15 zeros behind it. These are in massive quantities in the bloodstream of cancer patients, and there's a strong rationale for clearing these from the bloodstream prior to administering chemotherapy. One is they can actually export chemotherapy back out of tumor cells once it's been successfully delivered. The second point is the circulating presence correlates with treatment resistance. So the higher the presence, the higher circulating presence of tumor-derived exosomes, the less responsive people are to chemotherapy. In response, we developed ChemoPrep to reduce the bloodstream presence of tumor-derived exosomes prior to chemo administration. And our clinical objective corresponds with the initiative of FDA. It's called Project Optimus, to improve quality of life of cancer patients by reducing toxicity of cancer drugs with lower doses. And that's our clinical objective, is to maintain the effectiveness of chemotherapy with lower doses. And consider, what if we could maintain the same effectiveness or same delivery concentration to intended cancer cell targets with half a dose? Logically, that would cut toxicity in half for patients. ChemoPure, this is administered post-treatment. At some point in time after chemotherapy is administered, the chemotherapy that's remains left to circulate in the bloodstream is more likely to cause patient harm versus patient benefit. And we believe it would be beneficial and improve the quality of life of cancer patients if we could remove that. Next is immunotherapeutic antibodies. These are the top-selling drugs to treat cancer. These include your antibody drug conjugates, checkpoint inhibitors, monoclonal antibodies, and they're also the highest valued assets in global medicine based on 2023 and 24 M&A activity. In that regard, I might point out the acquisition, Pfizer's acquisition of Sigyn for $43 billion, and Merck's acquisition of Prometheus for $10.8 billion. Prometheus, you should know, did not have any market-approved antibodies. Everything was clinical stage, and yet that drove a $10.8 billion valuation. There's approximately 1,000 of these compounds in human studies, yet we are not aware of any strategy to improve the delivery of these particles, which is very poor. Based on peer-reviewed publications, less than 2% of doses reach their intended targets, and delivery is often intercepted by circulating decoys that display the binding site receptor of the antibody. So antibodies are known to have elegant specificity to bind to an antigen that's on the target of a cancer target. However, there's many other factors in the circulatory system that are expressing that same antigen receptor. And we intend to reduce the circulating presence of those. But based on this issue, these are considered to be revolutionary drugs in the treatment of cancer, but most patients still don't respond to therapy. And I'm going to give you an example of just how significant a drug decoy can be. If you look at rituximab, in this publication, it was demonstrated that half the dose, and rituximab is an anti-CD20 antibody, that half the dose was bound to CD20 exosomes 3 hours after drug infusion. We don't know what that was, 4 hours, 5 hours after, but just 3 hours after the infusion. And remember, Generally, less than 2% of these drugs get to their intended therapeutic targets. And if you think about the economics of that, consider a $10,000 drug dose is only providing maybe $200 in patient benefit. So our solution is the Immune Prep platform. This is a platform to incorporate a therapeutic antibody within a cartridge to sweep the decoys from circulation prior to the infusion of an antibody into a patient. We call this a reverse decoy mechanism. And this can be deployed with clinical stage antibodies. It could be used to repurpose therapeutic antibodies that perhaps didn't clear through clinical trials previously, and bring those back into the clinics for a second shot. But we believe the best application is for the use of market-cleared antibodies. And based on our historic communications with FDA, they have previously indicated that the ability to advance medical devices that incorporate approved biological agents have a significantly reduced clinical challenge as compared to trying to advance a non-approved biological agent inside a medical device. So outside of reducing the circulating burden of these decoys, the other aspect of the device is the simultaneous clearance of the antibody's therapeutic target to further enhance patient benefit without added drug toxicity. So in conclusion, at Sigyn Therapeutics, we're developing devices to optimize cancer therapies and address untreatable infectious disease disorders. Our endeavors started during the pandemic, and we now have an expansive therapeutic lineup based on 12 patents pending in the US and abroad. And in the field of extracorporeal blood purification, I believe our therapeutic lineup rivals that of any other organization. Chemo Prep, we designed to improve the delivery of chemotherapy to cancer cell targets. Chemo Pure, to extract off-target chemotherapy to reduce toxicity. Immune Prep, to improve the delivery of immunotherapeutic antibodies. And Sigyn Therapy, to address infectious disease disorders that are not treatable with drugs. And that represents a majority of the infectious disease disorders that affect humans. So with that said, I appreciate everybody's time. If you have further questions, please reach out to me at my contact. Email is below, jj@sigyntherapeutics.com. So Anna, with that, are there any questions that I can answer?
Yes, James, we do have some questions for you. Kinley says she likes the idea of improving the delivery. So if you can talk a little bit more about the problems with the current deliverability methods and why your method stands out.
Well, the current methods of delivery are very traditional. Patients go to infusion centers where therapeutic antibodies and chemotherapy is infused into their bloodstream. But there are circulating factors that are in high concentrations in the bloodstream that either interfere with the delivery of these products, such as in the case with chemotherapy, or in some cases, are expressing the actual targets of the immunotherapeutic antibodies, which remove them from the circulatory system and eliminate their possibility of providing patient benefit. And we think simply the incorporation of a device that could be easily delivered at the infusion center to reduce these factors prior to the subsequent infusion of these drugs into the patient would improve outcomes, would improve delivery, may allow for reduced doses to be administered, which improves patient quality of life. And in some cases with chemotherapy, it could reduce the long-term health consequences associated with chemotherapy delivery.
And so with regard to the dialysis machine, is the end product cleaner, or is it simply more efficient or faster? And what type of studies have been done about if it's cleaner or more efficient, if that's the case?
Yeah, we've demonstrated in vitro studies conducted by third parties that the device is very effective in removing viral pathogens, bacterial toxins, pro-inflammatory cytokines from human blood plasma. Subsequently, the device was used, and I'm talking specifically about Sigyn therapy. Subsequently, Sigyn therapy was deployed in mammal studies on dialysis machines without incident. I mentioned in terms of the application that could potentially emerge in xenotransplantation that we already have 8 treatment experiences in porcine models, porcine being pigs. So, we're confident in our products, and we have experience in this field. And as I mentioned, or I tried to reinforce at the outset, we designed each of these products from the ground up with the intent for them to provide the potential strategic competitive advantage that could be recognized by potentially a pharmaceutical organization or a dialysis organization based on results that could be occurring during clinical studies. And I think that's a major differentiator. Historically, extracorporeal blood purification devices have been perceived as salvage therapies. They have not been considered as therapies that have tremendous synergies to improve the benefit of established therapies. And I think that's a primary focus of our endeavors and why people should follow us closely.
And do you have any patents or IP that you can talk about?
I will tell you that I mentioned during the presentation, we currently have 12 patents pending in the United States and abroad. Again, we started the company from scratch in the middle of a pandemic. So I'm very proud of our IP position that we're developing. But each of these, the intent of each of these is to protect the use of our technologies in their applications, whether it's the removal of circulating factors that block or decoy the delivery of cancer drugs, or it's the removal of off-target chemotherapy that's left in the bloodstream of cancer patients, or the simultaneous clearance of inflammatory cytokines, bacterial toxins, and viral pathogens. Those are all claims made throughout our patent submissions.
And Jane wants you to explain in a little bit more detail what exactly your technology does. Question is, does it increase the compatibility of organs to humans? And is your device playing a part in this, or is it facilitating the transfer? If you could clarify that. So
there's 2 factors, and it's the primary goal is to reduce the circulating presence of pathogens. And this has been one of the gating factors for xenotransplantation, is that there are known identified pathogens in these animal species, but there's also unknown pathogens that could be transmitted, and there's not diagnostics for identifying unknown pathogens. So if you could treat the animal species prior to extraction of the organ, you could reduce the circulating presence or the accumulation of that pathogen in the donor organ before it's extracted for transplant into the patient. The same thing for an ESRD patient is the ability that during the transplant or even after the transplant, that you could treat the ESRD recipient, whether the organ's coming from an animal or whether it's coming from another human. You could treat the recipient to modulate down inflammatory factors that could cause rejection and pathogenic factors that could cause rejection. And in this application, the pathogenic factors are not only a concern from the donor organ species, but these patients are on major levels of immune suppressants to stop organ rejection. And as a result, they're at risk of having their own latent viral pathogens activated, which could cause a severe illness and lead to organ rejection.
Well, as always, really important work you're doing, James. Thank you so much. If you have any closing remarks for our viewers, please, the floor is yours.
No, I appreciate your interest in our endeavors. I think our work is extremely important. I think we're doing things in our field that are first in industry advances. We've done that previously. And I encourage you, if you have interest in the company or would like to schedule a discussion with me personally, again, my email's in the bottom left-hand corner of the last slide. It's jj@sigyntherapeutics.com. And I thank everyone for their time.
Thank you, James. We'll see you again in the near future.
Thank you, Anna.
All right, everyone, stay with us. We'll be right back.